Welcome to the Microscopists, a bite-sized bio podcast. Hosted by Peter O'Toole, sponsored by Zeiss Microscopy.
Today on the Microscopists, we sit down with the amazing Manu Prakash, and he explains the story behind the now legendary $1 paper microscope, the Foldscope. He tells us what inspired him to build a computer using bubbles, and shares why he has barnacles grown in his bathroom. All in this episode of the Microscopists. Hi, welcome to the Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by Manu Prakash from Stanford. Manu, how are you today?
Hi, everybody. I'm doing well, a little bit sleepy, a little tired, but excited to chat.
It's very difficult sometimes to know where to start with a guest because you're multifaceted. You're not known just for one aspect. But I guess what raised your profile for myself was a Foldscope. So actually, there'll be people who are listening who won't know what the Foldscope is. So would you like to describe what the Foldscope is to start with, and then we'll go down, find out why did you come up with the Foldscope?
Yeah, I've met people sometimes at a meeting and they say, "Oh, you're the paper microscope guy." I was like, "Sure." Yeah, no, the we invented the Foldscope almost 12, 13 years now. Uh, the challenge that I see in this world is stories of science are available, but experiences of science are very limited. I grew up in India thinking a lot about this and realizing that my own personal experience for science wasn't really a part of my learning sometimes. I explicitly remember the story of in my exam, we had to draw a microscope. And I don't know how many million kids in India are still doing that probably right now, but I had never seen a microscope. So how am I supposed to draw one? I'm supposed to draw one exactly as it was in a book, and the same perspective it was drawn in the book. It kind of stuck with me. And again, just when I started my lab, one of the things that I made an explicit decision, even before building microscopes, that I'm going to spend 50% of my time sharing the experience of science with other people. I can't write a paper and share that experience with anybody. I have to literally make tools and share those tools with others. And of course, we are on the microscope, as everybody knows. I talked to ChatGPT. It said it's top five tools that have changed science. I agree. And one of the important aspects of this was how do you share this experience? And we made a decision. And so, just to give people background, Foldscope is a paper microscope. It uses origami, micro optics to put together an instrument that, for a price point of $1 to $2, you can image submicron resolution. So, we had a joke in the lab, $1 single bacteria can we actually image? And after all kinds of tricks and failures and threads, we finally ended building one. And one of the key factors it's really is that the project has become less about the tool, but about the community. Currently, it's the largest amateur microscopy community in the world.
Wow.
2.8 million kids around the world, 6 million people reached so far. And it's a tool. I mean, I think the analogy that I often give is like pencils and pens. So, to a mathematician, what a pen or a pencil is, to me, a microscope is to a biologist. But if I was to say, "Oh, do you have one in your pocket?" And can you see immediately, the coffee that I'm drinking, or just thing that's sitting on my table, or some moss in my bathroom? Um, and one of the key aspects of this really has been is scaling the access to the community. So the project, I mean, of course, we've mapped microbial biodiversity with this tool, and the community is growing. But our goal has also been is putting these tools in hands of everyday people that are scientists at heart, and often would have such keen eye to observe, but have never had a chance to actually engage in science. So I want to tell less stories in science, but really share the aha moments that are genuine, that are created by people on their own.
So you're really enabling a lot of stuff. I'm just going to take you back. Did you say you got moss in your bathroom?
I do have all kinds of stuff growing in my bathroom. That is correct.
Is that by choice?
Uh,
Just checking it's by
I
choice.
have I have barnacles right now. I just got off a expedition in the Pacific Ocean and we ended up finding this massive buoy that was floating off and it was looked like plastic pollution, so we pulled it out. I mean, we were at the edge of the garbage patch and below it are these beautiful barnacles. So I'm trying to grow them in my bathroom right now. So there's a lot going on.
And you're married, is that correct?
I am married, that's correct.
And you've
We
got
have,
children.
yes, we do have kids as
What
well.
do they make of you trying to grow barnacles with moss and things like that in your bathroom?
We have two separate bathrooms. Uh, yeah,
Not anymore. You've got one
I
which is
often
now
like to not
common.
ask. That's correct. It's a little corner of a universe that I think I often feel sharing science is wearing your science and your passion on your sleeve. And, sometimes it rubs the wrong way. Most of the time, it makes people realize, that's interesting. And we leave it at that. I don't know what the kids think about that.
I, actually, I'm just reflecting for the first time. I remember the first Foldscope was actually one of my team, Graham Park, came into the office going, "Oh, look at this! This is amazing." He's got one on his mic on his desk still. But actually, I've just thought what you're doing. If I now think back to the very first microscope drawings, because people always used to draw. We didn't have
Yeah,
mobile phones that we could actually capture a digital image of what you're now looking at. Uh,
yeah,
and actually, the excitement that must have generated through the few people that could generate a lens from Van Leeuwenhoek, uh,
yeah.
Hook himself, Robert Hooke himself, and through actually now there's, as you say, millions, literally millions of
Hmm.
people who are having that
Hmm.
moment themselves. Because that
Hmm.
was still the reserve of the engineer, the physicist at the time, who had
Yeah,
arguably a hit scientist of some sort, but looking and
yeah,
just not even maybe trying to understand, but just documenting what they could see. Whereas now,
yeah.
wow, just think about
Yeah.
the number of scientists now looking down a microscope, discovering things that possibly no one else has seen before in some
Yeah,
cases.
and I think in what you said, there is a beautiful truth, which is we still know the tip of the iceberg of the microbial world. I mean, 99% of all species still remain unknown on our planet. And ironically, we're destroying the planet in many ways and forms, and we're possibly losing species faster than we can find them. So, in a context of this world, of that moment of discovery, those moments are still well and alive. Explorers are walking amongst us and legitimizing. And what's so important about this moment that you said, Leeuwenhoek was not a traditional scientist. He did not know Latin. He was not included in anything. And he was a fabric maker and, weaving threads, and he got interested in the small scale. And so some of his very first, I would argue, the most important discoveries would have been lost to the world unless he persevered and kept pushing the disbelief that existed. And so to me, and I think there is a dark side of all of this too, that he was very secretive. So one of the factors that I have felt, there's a very famous passage. I love his writings, and I'm working on a history book right now, which will come out soon, and kind of the historical context of all of this. And one of the big things that happened at that time, he explicitly said, and I'm quoting it because I'm hoping some young people are listening. He said, I can't find any young people that are actually interested in observations and pursuing science for the sake of science Everybody just wants to make money or make some quick bucks out of it. And so I am not going to teach this to anyone. That was not true. There were tons of young people. Everybody would have wanted to know. Yes, he got beaten by many other people with disbelief. I wish he had changed his attitude because of that decision for the next 150 years, nobody could make a microscope at his
No.
resolution. Cell theory could have been 100 years earlier. And it's so ironic that, I mean, I can understand he was outcast in many ways, but then it slowly, his capacity grew. But when it grew, he should have changed his mind and shared. And, I think the experience of Foldscope for us is that very first moment. The very first moment you explicitly remember looking down a barrel of a microscope. I have an explicit memory that's just ingrained in my eye. And often enough, just it is about the joy. I mean, first it's about the joy, and then you can figure out whether you're seeing something new or old and you're layering your experiences on top of it.
What was that image that you saw? What was that first image?
Mine is very complex because, ironically, we didn't have microscopes at school, and I was so obsessed at that time. I took my brother's eyeglasses and a cardboard sheet, kind of like a tube like this, and I just thought of microscopes as lenses and
Mhm.
tubes. And my brother was so upset, but I took his glasses from his lens and I put it together. And of course, I didn't see anything. The first time it was a blur because no, you don't just strap together some random lenses. And I broke his glasses for it and I got a beating for it. But it just was this sort of a blur. And then I remember very explicitly kind of coming back and I shared that in the school and kind of, then a teacher heard about it, and then there was in a biology classroom. We finally get a microscope, and uh, at that time we also I was obsessed with frogs. So the first image I really remember is frog skin. I was making skeletons of frogs at that time, and it was the frogs we would find in the neighborhood. And we would actually fix them in formalin and dissect them out and boil them in bleach to then build the jigsaw puzzle of the entire skeleton. So, I think it's that experience has ingrained in me the rawness of the living world. It was just some raw material. I mean, I would say we were the most undisciplined learners. Just learn by mucking around.
Yeah, trial and error, and yeah,
Trial and error completely. I mean, burning hands and doing things maybe as a kid we should not have done.
yeah, like playing with
Frankly,
behind.
now I look back at it and I actually think quite a lot about this notion of undisciplined learning, which just takes you places and you connect the dots, and then you have to build the analytical skills to understand the pieces. But if you don't see enough of a jigsaw puzzle, you really cannot take the leap.
So,
And
got a question thinking about your childhood.
yes.
A question I always ask is, can you remember the first job that you ever wanted to do? So maybe you
Oh,
were 8, 9, 10, I don't know.
yeah, I think that's a tough one. I found technology almost magical, and there was a photocopy machine that had opened in our town. It was a shop that you would go to and you would pay money, and it could replicate whatever you gave this person. So I remembered, I was like, oh, this is so cool. Uh, and the irony of this is that it's the Xerox machine, and it was the photocopier, and I was like, oh, I want to work and operate this machine. And to a certain sense, you might see the naivety in this. There is a person there just photocopying all day. But every time it photocopied, there was a light that you couldn't see what's going on. You just
Yeah.
see a flash of light. And I remember that as a kid, like, what the hell is inside this? And I remember actually later on, we went on these. Now there is a term for it. It's called dumpster diving. I didn't know that that's what it was called, but as a kid, we were dumpster divers. And we found parts of a broken photocopier. And it kind of was the first times like, wait a second, I had thought there would be something magical and it just had gears and plastic and electronics, like stuff that we knew. So I would say, yeah, that's my first job as a photocopy technician.
So I'm going to ask now, which might change tack a little bit.
Yes.
If you could do any job in the world, not your current job, for a
Mhm.
day or a week, is there another type of job that you would like to experience and get a feel for what it's really like?
You're saying what would I do?
Yeah, what would you like to sample? Not necessarily might not be what you want to be as a career. It might be just what would you like to feel? I think I've had people say politicians, astronauts. I don't think anyone said being a sports person. So, or
Mhm.
chefs. I think I've had in the kitchen to know what it's like. So, what sort of role would you like to experience and try?
I already wear many hats, and so it's kind of an interesting. I'm trying to go away from many of those hats because we spend time at sea and just being a sailor out in the ocean. Uh, Actually, I mean, it does. I don't know if it qualifies for a job. Nobody pays for it. But uh, yeah, I
Yeah.
think definitely sailing off for some period of time, but just on my own, probably a solo sailor.
So you've just come back from that. Before you left, did you realize that you would really enjoy that, or did you do it because you thought this is a good thing to do? I should be doing it. It will raise the profile of the problems that are out there. It will show that actually using the microscope, what you can enable, what you can realize, the invisible things that people don't see the pollution necessarily and the contaminants. Did you do it as a good thing to do and
Hmm.
not realize just how much you would love the open ocean, or were you already a fan of the open ocean?
Yeah, no, I mean, this was my 19th expedition in the open ocean. So
19th.
that's correct.
How
Yeah.
old are you?
Yeah. I'm turning 45 soon.
Good grief.
Yeah, yeah, no, I spent a lot of time at sea. And again, research expeditions is a key part of it. But it did happen. There was a point of time in which I explicitly made the decision that we're going to leave the terrestrial world and venture out. And at that moment, I had a gut feeling, which has turned out to be true now, which is, the window and the way that we have to look at the world. We wanted to turn that to the ocean, primarily because it's just the largest biosphere on our planet, and it's mostly dark. And ironically, that darkness is still in our knowledge and our understanding. And we realize that microscopy is one of the tools, although it just feels so vast. I mean, when I go on an ocean in a tiny boat, and the vastness of the ocean tells us, like, wait a second, there are trillions and trillions of these things right under my feet, and I'm trying to now observe one of them on a boat. I mean, it took us a while to build these instruments to survive. We now regularly take our home-built confocals to the sea. We would have the first time I was on a research vessel, everybody was surprised that, oh, look, there are microscopes for the first time. I was like, what? It doesn't make any sense to me. And so, I think it became kind of an obsession. And we have been trying with these tools to map the behavior, essentially, native behavior. Most of these species I cannot bring to the lab. They die within a few hours of bringing out of the water. The conditions have changed so dramatically. So we've been obsessed with mapping uh, a little bit like a behavioral atlas. So we're trying to now do physiology at sea, really just observe and be able to understand life cycles of these remarkable organisms. And, to a certain extent, life came from the ocean. I believe that. And so it's been It's been an obsession, and I think I would say it's again driven by just the vastness. I often feel that uh, when I think of a problem that's very hard to comprehend, it really attracts me.
It's probably a good time to mention now that
Hmm.
when I invited you, you said yes, but you were on I think you were on a trawler at the
That's
time
correct.
in the
That's
middle of the
correct.
ocean. The picture you said, actually, it just looked like a big trawler. I was expecting this nice ocean ship. Oh, it wasn't. Not at all.
No.
But I
Uh,
think, is this part of the ocean, the Observer film, or is that separate to the Observer film that's just come out?
yeah, no, I think this is separate from the Observer film. The Observer film is a philosophical reflection on the value of observation. And the film is just out now. And so if people want to host local screenings and threads. But the purpose of this was I had been teaching this class called Art of Observation, because just philosophically, I felt, what do I have to give to my students? I often feel, that's the most important thing that we all do many a times. And I felt to a certain extent, uh, my way of seeing the world or just the capacity to observe is something I do believe I can share. So I started teaching this class. And simultaneously, I met this phenomenal group and an incredible director who had been thinking about observation in his own ways. As a filmmaker, observation is a key part. And that conversation struck along, and I'm happy to be a small part of a much larger conversation around observation. And the film is just a delight. It's not just scientists, there are artists, poets, hunters, just teaching each other how to observe, why we observe, what does observation even mean? I mean, often enough, I study bioluminescence and we are out there and you see nothing. And you're like, yeah, this is what is, everybody's talking about this. And you wait and you close your eyes for 15 minutes, sit in the dark in a 4-degree C room, because much of what we keep, we bring from the deep ocean. It has to be kept alive at that temperature. And you open your eyes in 15 minutes of time, and then you see a totally new world.
Wow.
You are adapting yourself, you're changing. So just observation is not just about, oh, what can I see now? You have to prepare yourself for it. You have to prepare. I mean, as a human being, are we prepared to see what is there to see sometimes? And as a scientist, all your training is that preparation. When I think of a scientist training, it's dark adaptation to be ready
Oh,
for that moment, that you are sensitive to that faintest of signal. And it's ironic in many ways. I mean, the film really captures, it closes with an incredible blind recordist who records bird sounds around the world. And kind of, suddenly while watching the film, I just did a screening at Stanford yesterday for a crew and people on campus. And it dawned on me that we take the world for granted. We take our senses for granted. And once you can enhance those set of senses, either by tools like microscope or just sheer willpower, you start seeing and observing things that, I mean, this is a magical world, and we have often lost that touch in the
I saw the
rush.
pro. Yeah, I saw the promo, and I quite like the. From recollection, there was almost like your field square that you put on a piece of land
Correct.
and
That's
look, and I
correct.
quite like that. You're observing these snapshots, which also show that it's not showing you the world; it's just showing you how to observe that moment in time at that particular space, and
Yeah,
what's
yeah. And in microscopy, it's quite a lot of that when we keep observing, and we keep making these snapshots in the back of our head, and suddenly it clicks. Then you feel like, oh, I think I get it. And then we tell the world. But before that, much of it is just sitting around and uh, record the moments. It's important.
See, at the moment we've talked all about the the very much the sort of more social context and
Mm.
the frugal science and the hobbyist science and enabling it and bringing it to far more people, which is all nice and ideological. But I think what maybe hasn't come over yet is a
Mhm.
Foldscope is a physics design.
Mhm.
Right? So so actually, so let's just go back. Actually, your PhD, I think, was applied physics.
Yeah, applied physics. Yeah, I did a lot of continuum mechanics.
So you actually, what was your first degree in?
I got undergraduate in computer science. I hated sitting in front of a computer. So I
Okay,
have to do something else. I don't want to code, and I think I was right. Uh,
and that was in India,
that's correct. Yeah, I went to one of these IITs.
and then you switched to MIT in America. Then,
That's correct. Then I moved to MIT and I joined a lab that was working on quantum computing at that time. It was a combination of physics and
okay,
computing.
yeah. How did
And
you, before we talk about the science part, how did you find the cultural switch of moving to the US? Because the US is different to almost anywhere else in the world. So it doesn't matter
well,
where.
absolutely. Yeah. I mean, I would say I wasn't prepared for it. I didn't know what research was. In the Boston winter, I remember the first time I come out of my room, I was wearing sandals. In India, we really like wearing sandals. And I was like, oh my goodness, I'm gonna die. The first thing I did is bought some boots. Then I learned what black ice is, fell a couple of times. So, it just felt like it was viscerally devastating. It was like, I can't even stand on my feet. What the hell am I gonna do here? And then I remember in that same winter, like a little icicle fell and kind of hit my shoulder. It's like, oh my God, stuff is trying to kill me. So, those memories is what I remember. But then, slowly you kind of find your way. Culturally, it's interesting. It's, to a certain extent, universities, and this is very important of the importance of preserving a culture. Incredibly multicultural nature of our scientific institutions, which is clearly under attack now. It's not, American universities, I would argue, are not just American. They're a gift to the world if we want to preserve them that way. And they were made that way because those universities have been open to all kinds of ideas. So while all these cultures were there, I was playing in a Greek soccer team. I don't know, somehow I passed as a Greek, so they needed a soccer player. And I would go to a Lebanese lunch. I would be in a physics community, but spend time with the applied math group. And, it just, that's what has made many of these universities possible and taken it's, 20 years ago when I went to MIT. It was still like that. It's still like that today. The biggest thing I remembered was there are no bounds. You could do what your heart desires. And I think that was a big lesson for me. It's like I never got that lesson back before that sort of I'm in charge of my fate, but at the end of the day, I am on a driver's seat to choose what do I want to create in this world? What do I want to bring to this world, to life? And I almost recovered my childhood in grad school, I would say, because, India is a very tough academic place. Hundreds of millions of kids, they're applying for all these exams. And I had to go through these exams. So 16 hours on a bench, I remember I used to clock myself trying to prepare for these exams. I knew more math and chemistry coming into undergraduate than was the entire syllabus of MIT undergraduate. That's what we had to do to even qualify. But it strips something away, it strips away amongst us. You were talking about just pure hard work is not enough. This is a creative discipline. I often feel what we all do is far closer to art. We're artists of our own kind. You have to embrace that. And that was important. And I learned much of that back. It was all there. I relearned it So I kind of went closer to the roots of my childhood. And then my PhD ended up becoming, I didn't want to do quantum mechanics. It was not sure whether it was going to work or not. We're still struggling whether it's going to work or not. And sitting in the train one day, I mean, I'd been thinking about physics of computing and how biological and physical systems compute and what are the constraints. It dawned on me that you could actually build Turing machines using fluids. And actually, explicitly, I remember watching the subway map. If you ever see a subway map of Boston,
Hmm.
I saw in a subway map of Boston these little bubbles traveling like trains because they were representing trains. And kind of at that time, I had mathematically proved that it was not possible to build Turing machines using fluids because there is time does not occur in those equations, so they become linear. But you need nonlinearities for computing. And I remember on this train ride back home one night, it sort of resolved this puzzle for me, which was that, oh, I could use bubbles to make computers. And so MIT gave me a PhD for playing with bubbles. And I showed at that time that became what is called bubble logic now, that you can make a universal Turing machine using bubbles. That's sort of my fascination with kind of physics started there because I realized that there are things that you can create in this world that don't exist and will not exist unless you kind of forge them into existence.
I think if you go back to how you were with your undergraduate, and when you started, sorry, with your PhD, and you
Mhm,
started to learn again, you started to think and be
mhm,
more creative. And so the bubbles, and so your the bubble logic, that takes free thought, and it
mhm,
takes a moment of inspiration to
mhm,
come through.
mhm,
How now you're 45, you've got
mhm.
two children, you're married, you've got a research group, you have responsibilities to your team, yeah, your other family, not just your family at
Yes,
home.
yes,
Uh,
yes.
all the obligations like saying yes to me today, so thank you for that.
Mhm.
Do you find it harder to have those moments of inspiration?
Yeah, I think I have a very simple rule around this, which is I work in the field, so X percentage of the year I am mucking around in either the deep ocean or a swamp or a mountain looking for stuff. That's my moments of inspiration. And
Yeah,
then we spend years trying to, and again, Foldscope makes it very easy for me. We discovered hydrodynamic trigger waves. Um, it's a couple of years ago, we described how cells can talk to each other using pressure waves. And it's half a meter per second, so very close to electrical communication. It's the first example where you can use hydrodynamics and literally little shock waves to generate a massive trigger waves. And I discovered this organism in Lake Tahoe playing with my kids on a Foldscope. And so, and then it took us 2 or 3 years to figure out and understand and really unravel it. But the origin story of all the work that we've done and now on Spirostomum, many other groups have taken that on, is all actually goes back to that afternoon in Lake Tahoe when I was watching kids. And so, moments of inspiration, again, going back to the theme of observation, you have to be ready for it. And it is one of the fundamental blocks. Of course, many of our analytical tools are very important. Our deep knowledge in a field is important. But a key element is that element of surprise, is the element of observation. And that's remained, I think if I stop doing that, I will stop being a scientist. I will do something else because, and again, I think my job, going back to my duties and for my students, the first thing I do when a new postdoc or a new student arrives, I take them out to the field. So just 2 weeks ago, I had a fresh student arrived as a rotation student, but he was on a boat with me for 2 weeks at sea in the middle of the ocean. He didn't know he was signing up for it. And it was a little bit of a, okay, let's find out just what the world has to offer. And these types of moments are important. We have to share them. It often goes back to the experience because no matter what, if I can see and if I can share a way to ask questions, answers will come. They eventually always come. Even if we don't find the answer, somebody else will find the answers. I get excited about questions, and I think to a certain extent, it's on us as a duty and a responsibility to teach the way, new ways of thinking in science. It's not the only way, but often enough, that's my biggest responsibility that I take. And so I share my time out in the field with my kids and my students. And I'm making it sound easy. It's not easy. There's lots of sacrifices. I wish I could be there for the birthday of my kids, and I was not. And it sucks. It just
How old
hurts.
are they?
They just turned 9.
Oh, twins.
I have twins. Yes, yes.
Ah,
Yeah, but it just, I mean, I struggle with this. Uh, I've just kind of agreed on this fact that none of us are ideal in any way and form, and we have to live with that. And I think in certain sense, I feel the sharing our joy of the deep joy for science. I don't, if we don't do that, it's not going to survive. As a discipline of the true curiosity-driven science will not survive in society unless we pass it on, unless we make it important, unless we put an anchor on it.
are your children really keen on microscopy or looking down the Foldscope, or are they, oh, Dad,
Hey,
you have
hey.
to?
Yeah, yeah, both. It depends on the mood. Yeah, I think it's a little bit about building habits. So when we go on a picnic and we're out, I'm just doing what I would like to do,
Mhm.
and something rubs off on people, and it's often, um, I think I mean, I'm teaching and running Foldscope workshops for thousands of kids around the world all the time. It's a very large community, so I am in. Um, we run programs. Just a couple months ago, we were in Dharavi. It's the largest slum in India, running Foldscope workshops. And the community has grown rapidly. So often enough, I kind of think about, uh, I would just much rather share this process with anybody who wants to, and it sticks with somebody. And it's also, sometimes. The analogy to all of this is that the aha moment, a genuine aha moment, might not occur in that first moment of contact. It's when you take this object and you put it away and it's on your shelf, and at a much later time, you suddenly were puzzled. Nobody was looking, you were just working on your homework assignment, and suddenly you looked at something, almost as a procrastination, and then it dawns on you. So, learning and true moments of inspiration, you cannot engineer them. They occur spontaneously. So we have to leave space. So often enough with Foldscope, there is a rule like Fight Club, there is a rule of a Foldscope club. Once you share the tool, you pass it on, you give it to the person, you let them muck around, you give them that same freedom.
I say my moments of inspiration are quite often. So I don't
Yeah,
drink a lot of caffeine, but
yeah,
when I do,
yeah,
yeah, the mind is far more open.
yeah, yeah. No, and I think it's personal too, right? It's our private spaces, it's our ideas are very vulnerable. Like, I often think ideas are very shy. Even in my own head, some of the most exciting ideas are hiding behind some of the most common ideas, and they don't like coming out that easily. And to a certain extent, it's a function of how much do we believe in something. Will we go out on a limb? Because, going out on a limb is hard because you fall flat and we are used to it. And so in some sense, that's why private time thinking is critical and thinking with your tools, and it doesn't happen in isolation. So, what coffee does to you, microscopy does to me out in the middle of nowhere. I would be any ecosystem, any place, any object. I want to know what's down below.
Yeah, but I bet you're drinking coffee on the ship.
Oh, I am massively drinking
Yeah, so
coffee.
you need both to go together.
Yes.
Okay, Manu, I'm going to ask some quick fire questions just to
Yes,
check
go
back
for
in
it.
there. So, are you an early bird as he drinks coffee, an early bird or night owl?
Um, I'm a night owl,
Okay.
but I do have to wake early to I like going to school with kids.
Yeah,
So,
you do the
but
school run. Uh,
yeah, so it's kind of changed me a little bit, but when I'm in the field, I am the nightest of night owl. I'll meet everybody for breakfast and then I'll go to bed.
okay. Are you just going to bed at that point?
That's correct. That's
Oh,
correct.
crikey. Okay.
Yeah.
That's not a night owl. That's
Yeah, I'm
diurnal.
on a night shift. No, I
PC or
like
Mac?
my, yeah, I like the nighttime.
PC or Mac?
Oh, I didn't know what a Mac was a long time ago, and then I got into it, and now it's only Macs.
Okay. I've got a feeling I know the answer to this one. McDonald's or Burger King?
Ooh, I think I let my kids often choose, and it's often McDonald's.
Okay. I thought I was going to get none of those. That's cool.
No,
Uh,
no, I think we compromise. We go on long road trips with the family, and we stop wherever we can.
there's nothing wrong with a cheeky McDonald's. Tea or coffee?
Oh, coffee for sure.
Uh, Beer or wine?
Probably none. Yeah.
Chocolate or cheese?
Hmm. Yeah, maybe chocolate.
Okay. What is your favorite food? So actually, if you were to be taken out, what is it that you'd most like to be served? What's your favorite food?
You know, I left India. I mean, I go back often enough, but I miss India a lot, and I miss the street food in India. So my favorite food would be is quite literally street food in
Yeah.
India. And unfortunately, the way microbiomes work, I can't have it anymore because I
Oh,
cannot tolerate it and like I get sick sometimes. So I'm very cautious when I do eat street food in India, but that's what I miss.
okay. Uh, is there any food that you do not like?
No, I eat everything. I eat insects. I eat whatever I can find. I will try everything once.
Would you rather eat in or eat out?
Yeah, I think that's tough. It depends on where we are. When we are in new countries and new places and just traveling with family or with my lab, I like eating out. I
Okay.
want to try and be as close to whichever culture we're trying to experience.
But at home, I presume, therefore, you like to eat in.
That's correct. Yes, yes,
And
at home.
do you prefer to cook or wash?
Uh, I, I, many times I'm not even at home. So I'm neither cooking or washing sometimes, but when I am at home, I'm washing.
Okay. So I
Yes.
take it your partner's the better cook.
Oh, yes. I think we have this thread around. We just eat whatever we can
Hmm.
for a little bit of time.
Book or TV?
Uh, I have a sticker on my Mac that says that I'm obsessed with buying books. So it has to be books.
Okay. And what sort of genre? What are you reading, or what sort of books are you buying?
I really like just mostly history and biographies.
Okay.
I like understanding people.
Do you have a favorite film?
Ooh, that's a tough one. There are so many of them. Um, I think I like international films most often, and especially films that kind of are hidden in corner. I mean, I love Totoro. Is in the common themes, Totoro would come to mind.
Yeah.
But there's an Iranian film about a kid with one shoe. It's actually also quite famous now. It's one of my favorite films, where two kids are trying to go to school, but they only have one pair of shoes.
You do realise you failed the answer because you really should have said The Observer.
No, 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 I can't say that. I mean, it's a beautiful film. Yeah, but I wanted people to judge it for
No,
themselves.
I am joking, but the director won't be pleased. Do you have
No,
a
no,
favorite?
I think the film is a very honest expression of people, and it's so whimsical in many ways too.
Mhm.
But again, just like we know science films have a challenging time, and the big thing that we've been thinking quite a lot about is just the raw honesty in that film. Some people will deeply appreciate it, and other people will be confused. It's like, oh, this is not the picture of scientists that we thought, but it's okay.
What about music? What sort of music do you like listening to?
So I am musically challenged, and so it's basically whatever the kids want to play. That happens to be Taylor Swift in this season, or it's really the kids deciding. It's growing up, I don't think we had much access to music, unfortunately, which was sad. Now I look back and see, oh, I wish that was there, but didn't have that.
Favorite color?
Mm, right now it's black. It changes quite a lot, coming from the deep sea and realizing the depth of blackness, the blackest of the blacks.
Did a
And
physicist just say black's a color?
yeah, yeah, I would say the blackest of blacks.
That's a cool answer. Uh, Physical biology or frugal science?
Ooh, that's a very tough one.
That's a naughty question.
Yes, I think I would argue that I discover a lot of physical biology with my frugal science tools. So as we keep growing the frugal science tools, there is an infinite number of physical biology questions that we have discovered. Every single paper, I mean, the lacquer paper we did recently on origami in single cells was again mucking around in that pond with a Foldscope. It's a pond close by Stanford campus. I went on Google Maps, say, okay, I want a water body. We found a little blue dot on it needed to be 15 minutes away because I didn't have that much time that day, so it needed to be quick. Went out, the first drop I look, and boom. I mean, this is something that I had searched for this animal organism cell for 15 years and I didn't find it. I didn't find it in the most pristine of jungles and places and then right in this kind of a sewage fed pond, I found the most beautiful thing I was looking for. And so these things are deeply connected.
I'm just thinking out loud now. So this is kind of going a bit off on a tangent of what the microscopy is usually about. With
Yes.
all these images being captured of organisms that are probably not yet described,
Hmm.
are they being catalogued anywhere?
Yeah, 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 yeah. I mean, we are very adamant about it. So the community entirely, if any of you go on the website called microcosmos.foldscope.com, it's a massive repository of just curious people documenting what they do. We don't force it, but now there is an app, so it auto logs itself. And then we have many taxonomists. And there's a new program that I'm excited about. We haven't publicly announced it, but I can just describe it briefly. Is we're announcing a program. I mean, first of all, people have discovered totally new species using Foldscopes. They've been now named, sequenced, and a new program that we are announcing in partnership with sequencing partners is just like Reddit threads. If that sets of images, many people say, oh, we can't identify what it is. You upvote it, and every day, every week, we choose one sample for sequencing, but run completely by the community. And the purpose of that is to then start giving IDs and molecular IDs to many of these sets of samples itself. So a lot of people in the community are excited about this because it also connects low tech, high tech in many ways. And every post has a DOI number. So then it sort of allows us to anchor many of the ideas into. You can go back and cite an observation and then build an entire framework and sets of questions around it.
So it's a huge citizen science initiative. So, the Foldscope itself is one thing, which is your frugal science, but actually that has mobilized a
Mhm.
whole lot of citizen scientists
Yeah,
generating this data bank of information. So my
yeah.
next question is there machine learning that is actually identifying the same species or commonalities and then bridging so from different, because you've obviously got the location of each of these species where they're
Yeah,
found.
the ecological part is there. Yeah, so I think the ML, there is two layers that have we've built, and there's actually two tools we have built around that. One is for the aquatic world, which is called Planktoscope. So Planktoscope is a brother of Foldscope or a sister of Foldscope in some way. Now that automates, it kind of moves from $1 to $100 kind of regime, but it automates and is a flow through system that will image everything. And then we build classification pipelines. So then you are imaging everything in a given volume of water. Foldscope now with the app, there is an ML layer to it, and it'll classify some of the most common bugs. And one of the things we're doing right now is we're building with the data set that we have, we're starting to build the ID tools. It's quite hard in microscopy because, again, context is different,
And
different cell
orientation and
phones,
location.
orientations. So we're actually working on video models. Video models also include behavior. So behavior is an important classifier in many ways. And often when I am classifying, I'm actually looking at behavior as the first cue.
Yeah.
Like just the right way the ciliate will move gives me a clue. It's like, okay, I know the it's not using cilia in the same way or the other. And that sort of drives these models. But yeah, we're very excited about this because for the first time now we can match and people can go from, oh, what the hell is that? To having some rational idea about what it could be, which gets people very excited.
We've got I guess I haven't really discussed, we haven't really published much in this area. We've actually got ourselves at York, along with the Broad Institute,
Mhm,
Sheffield, and some collaborators in South Africa, Brazil, Nigeria, looking to develop microscopies, but in the lab microscopes
mhm,
for very low cost.
mhm.
So using things like the open flexure
Yeah, yeah,
microscope,
yes,
biolights.
yes.
But as you say, it's the analysis that's key because actually
Mhm.
there's a lot of data, but actually it's very hard to interpret at the moment because there aren't the tools, the machine learning tools.
Yeah,
And actually, Laura Wiggins, who's a PhD, flying now by herself with something called self phenotyping, and it
yes.
is actually the protein behavior. Sorry, protein, the cell
Yeah, yeah, cell
behavior
behavior.
that is
Yeah,
one of the biggest differentiators. And even within a monoculture,
yeah,
looking at those behavioral differences to understand what will resist a drug, what will not resist a drug. And we're trying to roll that out at the moment to make it available. So once, yeah, I can just envisage, people in the low middle
yeah.
income countries being inspired by the Foldscope,
Mhm,
going to universities and then not having a microscope. Because
mhm.
actually there's many that still don't have microscopes.
Yeah.
It sounds daft from the Western world and when our high schools have them and stuff that some
No,
universities
that's the
don't. So
reality. No, 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 absolutely. And I think again, one of the key things in that space is I'm a big fan of open flexure and many platforms. It's a little bit like a valley of wildflowers. We have to have all flowers bloom. And then another sets of tools that if people love microscopy, I don't know if people have heard of Cephla. I've just started my third company now, which is called Cephla, and we basically are now manufacturing squids. Squid was another platform, and Octopi is another imaging platform. And now it kind of goes, so the sequence is foldscopes, planktoscopes, and then squids. And squids are really about automation. And one of the key factors is it's completely open source. All of these sets of tools people can build, all software is open. But now, I mean, we build kind of in this context, confocals, we can do optical pool screening, we can do in situ sequencing, codecs in single platforms, but that have a capacity to be modular. You
Yeah.
can start modifying them. And one of the key sets of ideas in this space is really we need a continuum, just like what a pipette became such a common tool on everybody's bench.
Mhm.
We need microscopes to be in that same vein. Otherwise, we're going to miss out.
Yeah, we're
In
going to
the
miss.
old time, the data used to be an issue. Now, data is not that big an issue. We can handle it.
I think the biggest issue, I would say, from what we're looking at, isn't just they're generating data.
Mhm.
Or hopefully, if we put the microscopes in the hands, they can generate the data, but they need to be electrically safe, as in
Yes.
not to be able to withstand power cuts, is what I
Yeah.
mean. So actually
Yeah.
making
Yeah.
them
Yeah.
low
So
energy,
Octopi,
really.
for example, the paper that we just posted, it's called Octopi 2.0 now. Um, it's one of the key things that you pointed out now, which we had to deal with. It's a malaria diagnostic tool. And every time we were out in the field, we would get power outages.
Yeah.
So we had to build an inference engine for ML on the tool that could handle 8 to 10 hours with battery power alone. And it was very important because in a diagnostic setting, power goes in and out in a primary health center. And one thing that I'm really excited about is finally now roughly around 25 of them are in research institutes and clinical trials and validation trials. And it's become really the hub of a massive number of, I think we have around 7 to 10 different diseases that the ML models have been trained for on an octopi. And so I'm calling it the App Store for diagnostics because and it's completely open as an architecture, so you might say have an expertise in leishmaniasis, you're a clinician, you can annotate the sets of images, you can run a clinical trial, you can build the app that can run on the platform. So we're also starting to build this in a manner that the hardware is stable, has regulatory approval separately, and we open up the app store. And I
And
think
so
it's been a really fascinating thing because so many people are deeply excited, but they have the clinical expertise that is critical when it comes to these ML models. Because, on one hand, in science, we have a hit rate, okay, 90% of the time you got it right. It's okay. We can, but when it's human patients, it gets very hard. And that last squeezing the last percentages is really about recruiting the best clinicians in the world.
interesting. The leash is something is one of our targets with our Brazilian
Yeah. Yeah.
partners at this point, and it is the ML that is actually you can do on here and not
Yeah.
have to be dependent on the cloud because so much has gone to the cloud. Again,
Uh,
in these resource where the resources are more
yeah.
limited, it is not reliable. So with Beth Simony is really focused with our
Yeah.
Pixamy. So it's quite
Yeah.
a good team from that. We've got 4 minutes left.
Yes.
Well, way too fast. Who has been your inspirations?
Oh, just so many people. I think I often think about uh, everyday people that I meet. I often, in most of my life, whenever I've felt this, that uh, everybody's job is equally hard. So, the person who's helping as a technician on a boat, and my life is depends on him, and I'm out doing an expedition. Um, it's just, I think I enjoy this idea of, I was telling you about history of people. It's everyday people that just love what they do is, I enjoy that. To me, we as a species are storytellers, so we can always put spotlight on some people. But to really understand what happened, you really have to understand the fabric that was around and why something happened, why the sets of chances. So I often like, whenever I'm kind of struggling, I'm often thinking a little bit about, wait a second, let me put myself in the shoe of this everyday person that I know that works right here.
Which is cool, actually, which really chimes with the fact and go back to the title of the film, The Observer, and the
Yeah.
microscope and observing. what hasn't
We're
been.
all scientists. I
Same with
think we
people.
all have equal capabilities and capacities. Just some of us get more obsessed with it.
Have you
And
had any? What has been the most difficult time in your career?
it's an interesting question. I've career is a I'll answer this much more from a current challenges of what's going on.
Mhm.
I feel the first time in my life I feel science is under attack explicitly. I've
This is
never
the
felt this way.
USA.
I'm talking about in the US, I'm talking about the last 3 months of this current administration. I've dedicated my life to working on global health, climate related problems, basic science, which is raw curiosity. You don't know where the wonder leads you. And I've spent a significant amount of my time sharing and building capacity for people to both enjoy that and in all fractions of life, not biased towards one or the other. And it pains me so much that in a short period of time, people that have absolutely no understanding of what has made science the engine of every economic progress. I mean, our scientific institutions here in the United States are an envy to the world. Uh, the joy that it has brought to people in communities, the kind of jobs that it's brought to sets of community, and the fastest destruction that I have seen in not just the infrastructure. I mean, USAID and the President's Malaria Initiative. What a foolish thing to do to say that, oh, we're going to devastate this entire capacity in the world for people to engage, not realizing that if you cut all programs around malaria, we're going to be swamped with just these sets of diseases and all of the more deadlier diseases that have even a higher index of transmission, like Ebola and Lassa, will all be unchecked. So, in some sense, it's even from a selfish point of view, I find it. So, I feel what's happening currently, I feel the most agonized and the most I think it's we as scientists now, there is clarity that's emerging in the last 3 months. And so people are starting to think about, we're still in a very reactive preservation mode. We have no clue what's coming. But it's been very difficult to think about. And again, I don't want to tie it to career. Who cares whether any one of us as individual make it through the turmoil, or it doesn't. I don't know, we'll find ways as individuals. But as a collective, we have broken something. The kind of fear that I am seeing in students' eyes about, oh, wait a second, whether my visa is going to be revoked or not. It's just such a shameful thing for us to be even dealing with. Like, on one hand, we talk about inspiration and these sets of values, and on the other hand, we're literally driving fear in hearts of individuals. And just
Yeah,
deeply troubling times from my point of view.
this time last, literally this time last week, Sir Paul Nurse, who's the
Yeah,
incoming president of the Royal Society, was talking very carefully, talking
yeah, yeah,
uh, because obviously you have Elon that is seen to be driving a lot of this,
yeah,
and it will be interesting to see where it ends up. And I'm ever the optimist. I must say, I might be skeptical, but I'm quite an optimistic skeptic, if that makes sense.
mhm, mhm,
So I always question, but I'm always optimistic. It isn't great times. That's clear. I don't think anyone would deny it. I think if you ask Elon Musk, he would say it's not easy times. He will say it as a medicine or whatever. Hopefully, the scientific community
mhm,
will come ever closer together and be
mhm,
less competitive against itself
mhm,
and even more collegiate
mhm,
to get through this. And as the optimist,
mhm,
once we ride out the other side of this, we'll ride out strongly in
mhm,
a united fashion.
mhm, mhm.
Uh,
Yeah,
that
that's my hope. I think, I mean, in some sense, when COVID happened, we saw signs of that across the scientific community. And, I'm not, I know we will come through this whichever way and we'll rebuild. Uh, but there is a sense of loss that happens. The young people are caught in this. Uh, Students, postdocs that are in faculty job markets right now, they're not going to get that moment back. They're not going to get those chances back. The fellowships that are being canceled, the NSF support that's just been pulled under the rug. So I think the challenge around this is longer term, we have always found ways, and retrospectively, we can always come and people will write their own histories from that time. And nobody really knows, you're right, what will end up happening. And lots of good things might come out of it too. But it didn't
Yeah,
have to
yeah,
be this
there's
way.
a
That's
lot
the
of it.
problem. But it comes at a cost that's too heavy in my mind. It comes at a cost of young people's careers and dreams, and the wonder and joy that we have built the scientific enterprise on cannot be preserved because it feels hypocritical in many ways. On uh, Pulling the rug on science in this way and form, and doing it in a sense that's really driven fear in many people's hearts. That's really what bothers me. There is
Yeah.
an emotional side to it. It's not just the rational side. And we do the numbers, who knows what comes out. Maybe this will lead to a overwhelming support for science, and 5 years from now, we would be in a better place. But what is lost?
But I would worry that the lay public who are not scientists necessarily, the non-scientists, possibly don't understand the impact and the future potential impact that science will deliver. And if we look around us, everything in my office now, I'm talking to you on a
Mhm,
laptop with a light, with a microphone, with my phone next to me,
mhm,
drinking a coffee that scientists have analyzed, worked out how to preserve, keep, refresh, ecologically reduce its footprint. So
mhm.
much science is all around us, and I think we take it for granted, or it can be taken for granted.
It has been, yeah,
It
yeah.
will slow the development of science up.
Mhm.
A bit like you say with Van Leeuwenhoek. If we go
Yeah,
right back to that point, and you said how he
yeah.
didn't say how it's done, and it's delayed cell
Mhm,
biology for
mhm.
100,
Yeah.
150 years.
Mhm,
This impact could have a similar
mhm,
fundamental impact of delaying those discoveries that, if it wasn't for that, we would never have had a vaccine for COVID.
mhm,
And this could delay the next big find that could
mhm.
prevent the antimicrobial resistance, the next way to solve antimicrobial resistance, which is a big potential problem. Anyway, I realised we're up over the hour, and it's about. I hope you mentioned earlier how how many say you could put a spotlight on anyone, and I hope that this has helped put a spotlight on yourself, Manu, and to people to get to know you better. Uh,
No, I'm happy to chat. It gives me a lot of joy sometimes to just share philosophical ramblings, even if they're ramblings, because there might be nuggets of truth that other people find useful.
and I realise I'm going complete. My sunlight is coming through. It's that day.
Yeah,
This,
telling us that we are
you
in two
said you like
different
black.
time zones.
I'm going very black on one side. Uh,
I love the fact that we can see the sun though.
yeah, Manu, thank you so much for joining me today. I will just give a shout out. The citizen science side, there was a lovely one with Lucy Collinson talking about citizen science, about the analyzing data. There was one with Chris Lintott, uh, who incredibly isn't a biologist, but set up Zooniverse that is now not just for looking at penguins on icebergs and in Antarctica, but also for scientific analysis of data. And everything you talked about today, man, it really chimes with all those different programs as well. And I realize time is precious. And thank you for joining me today. And thank you everyone who's listened and watched. But Manu, carry on inspiring and carry
Thank
on
you so much. And
innovating.
if anything that I said, if people find relevant, I'm around. Reach out. Many of the programs that we run are literally owned by communities. So if any of you want to start your own Foldscope community or your squid and octopi communities or your planktoscope communities, you don't need anybody's permission. We've built all of these tools in the open. I would love for people listening to kind of take ownership of these programs and carry on. Take them into hearts of places where your heart is.
Manu, thank you very much for joining me today. Thank you, everyone.
Bye, Peter.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.